Good morning, ladies and gentlemen. Welcome to this public lecture, What is Life, by Sir Paul Maximus, a Nobel laureate in physiology or medicine and renowned geneticist and cell biologist. My name is Joyce, a Year 5 student from the Faculty of Business Administration, and it is my honor to be your MC today. First of all, please extend our very warm welcome to Sir Partners for coming to CUHK from UK to exchange with us on the question of what is life. Sir Partners is known for his groundbreaking research on the eukaryotic cell cycle. His book, What is Life, has been published in 22 countries, exploring fundamental questions about biology. We have an exciting program today. On this intriguing topic, Sir Partners will first delve into the question, what is life, through the lens of 5 fundamental concepts of biology to help us deep dive in an exploration of some original forms of our biological being. Following his presentation, we will open questions to the floor for a Q&A section to be moderated by Professor Yuk-ming, CUHK's Vice-Chancellor and President, who is a pioneer in liquid biopsy and also known as the father of non-invasive prenatal testing. Without further ado, let's welcome Professor Low to deliver a welcoming address. Professor Low, please.
Sir Partners, Dame Julie Maxton, Professor John Chai, distinguished guests, colleagues, students, ladies and gentlemen. Good afternoon and welcome. So thank you very much for joining us at the Chinese University of Hong Kong for this public lecture by Sir Partners, a Nobel laureate and a most esteemed friend of CUHK, who has made extraordinary contribution to global science. First of all, I'd like to extend my heartfelt gratitude to Sir Paul for gracing us with his presence on campus. Now, Sir Paul, we're deeply honored to welcome you back today as a member of the university since the conferment on an honored doctorate upon your good self in 2024. Sir Paul's reputation speaks for itself in the scientific community. He is a distinguished geneticist and cell biologist. He won the Nobel Prize jointly with 2 other scientists in 2001 for the discovery of key regulators of the cell cycle. Over the past 30 years, he has held many significant leadership roles. For example, he is the honorary life president of the Francis Crick Institute in London, which is Europe's largest biomedical research facility, the Chancellor of the University of Bristol. From 2010 to 2015, he was the president of the Royal Society, which is the oldest scientific academy in continuing existence in the world, and has assumed presidency again in December last year. He is actually the only individual who has held this position twice in the 365 years history of the Royal Society. Now, he has also previously served as a chief executive of Cancer Research UK and the president of Rockefeller University. Sir Paul's groundbreaking research and remarkable commitment to science, medicine, education, and humanity have won him numerous prestigious awards, including the Lasker Award, the Gardner Award, the Albert Einstein World Award of Science, and the Royal Society Copley and Royal Medals, as well as honors such as a knighthood, an appointment to the Order of Companions of Honor, and the Order of Merit from the UK, and the Order of the Rising Sun from Japan, amongst other accolades. Now, throughout my own academic journey, I've been inspired by many great leaders and prominent scientists, with Paul amongst the foremost. Actually, when I was elected as a Fellow Royal Society back in 2011, I had the honor of having Sir Paul presiding over the proceedings. I'm therefore particularly looking forward to Sir Paul's lecture today. Now, in today's lecture, Sir Paul will share with us his thoughts about the question, what is life? Now, this is a question which has been asked by scientists and non-scientists alike over the centuries. So, ladies and gentlemen, without further ado, please join me in extending a warm welcome to Sir Partners, who will take us on an illuminating journey to seek answers to the intriguing question, what is life? Sir Paul, please.
Thank you, Professor Low.
Well, thank you for that very nice introduction. It's a pleasure to be in Hong Kong. It's a pleasure to be back here in the university. I'm delighted to be speaking to you. I'm going to talk about one of the most fundamental ideas in biology. What is life? It's a question easy to ask. I have to be honest with you, not so easy to answer. And you're not going to get quite a dictionary-type answer to the question. But I hope you will learn something in the journey we're going to do together. There is an extraordinary diversity of life on our planet. All around us are living things, bacteria, fungi, plants, animals, and of course, ourselves, human beings. So the question is, what is it that links all these different life forms together? It's quite a long time ago, but I think it was a butterfly, like this one you see here on this slide, that first started me thinking seriously about this question. It was early spring. I was only around 12 years old, and I was sitting in our garden in northwest London when a quivering yellow butterfly flew over the fence. It turned, it hovered, it briefly settled, then a shadow disturbed it, it took flight, disappearing over the opposite fence. A very simple encounter. But that intricate, perfectly formed brimstone yellow butterfly, that's what it is, made me think, because it was both utterly different to me, of course, but also somehow familiar. Like me, it was obviously alive. It could move, it could sense, it could respond. It seemed full of purpose. I found myself wondering, what does it really mean to be alive? That is, of course, what is life? As you just heard, this question has been asked by others over the years. For example, this is the title page of a book, one of several written by different authors, called What is Life? This one was written in 1944 by the famous physicist Erwin Schrödinger. His book was influential, including for me when I first read it now half a century ago. He had a particular interest in the transmission of heredity. I'm going to be somewhat broader than that. My approach is going to consider 5 of what I think are amongst the most important ideas of biology, describing where they come from, explaining what they are, then pulling them together to develop principles fundamental to life. And that, I hope, will inform you what life is. The first of these is the concept of the cell. The cell is the basic structural and functional unit of life. It is life's atom, if you like. Cells were discovered by Robert Hooke in 1665 in the city I live, which is Oxford in England. He used a microscope like the one shown here on the left, and he took a razor and he cut slithers of cork and examined them under his microscope. And what he saw were neat arrays of boxes. You see the image in black and white that he actually drew. And he called them cells after the Latin word for cella, for small cubicle. It's published that in a book he published called Micrographia. On the right there, you see in the yellow image is what we would see today with a scanning electron micrograph. To be quite honest, it's not much better than what Hooke drew in 1665. A few years later, the Dutch draper Antony van Leeuwenhoek, working in Delft, scraped between his teeth and discovered single-celled life, bacteria. He sent this drawing to the Royal Society in London in the 1680s. I've actually held it. It has this rather, I find it, charming image of pictures of bacteria. And if you look at Fig B, you see the bacterium was obviously swimming about and did a loop-the-loop going from B to C to D. He was frankly rather disturbed by this discovery of microorganisms living between his teeth, because he was very proud of his dental hygiene, and he certainly found it disturbing to find life in his teeth. He was also a close neighbour in Delft to the painter Vermeer, who painted very unusually for a 17th century artist 2 pictures, idealized pictures of a scientist. This is one of them. There's one in Frankfurt, one in the Louvre in Paris. I like to think that Vermeer was motivated by Leeuwenhoek, and in some sense this is an ideal picture of Leeuwenhoek. I have to admit to you there is no good evidence for this speculation, except when Vermeer died, Leeuwenhoek was actually the executor of his will. So there was a connection. It's not completely impossible. Over the next 2 centuries, it became clear that all living organisms consist either of a single cell or a collection of cells. This conclusion was summarized by the German zoologist Theodor Schwamm, who in 1839, in German of course, wrote, all organisms are composed of essentially like parts, namely of cells. So, but as well as cells being the basic structural unit of life, which is what's described here, cells are also the basic functional unit of life. There is a distinction there. This was summarized by another German scientist, Rudolf Virchow, who is actually the originator of pathology in the mid-19th century. And he quoted again in German, translated here, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. That's what I mean, the basic functional as well as structural. He also used the term, the sentence, omnis cellula e cellula, Latin for all cells come from cells. Look at the mistake there, form cells. I keep forgetting to correct it every time I give this talk. He wasn't the first to use that. It was used earlier, 20, 30 years. But what it, of course, deals with is the origin of cells, which arise through the division of pre-existing cells. That was, in the mid-19th century, not generally what was thought. It was thought by many that cells spontaneously arose. So cells are interesting, basic structural and functional unit of life. But if I failed to convince you of that, let me show you this picture. This is a mammalian egg being fertilized by sperm, and I want to remind every one of you in this room that all of you were once looked like this. All of you were once a single cell. If you're still not interested, there's 2 exits actually, one over there and one over there. Cells are considered to be the simplest entities which unambiguously exhibit the characteristics of life. I use the word unambiguously. We'll touch upon that when we get upon more intermediate situations a bit later in my talk. And this means, of course, that cells and understanding cells are key to understanding life. And it allows us to focus on what is really important. So they're going to figure prominently in this talk. This is a modern picture of a cell, artificially colored. What are cells? They are bounded physical entities separated from their environment, surrounded by a semi-permeable membrane, which separates them from their surroundings, but allows communications with those surroundings. This separation allows order to be built up inside cells at the expense of increasing disorder outside cells. And I emphasize that because in this way, life does not contravene the second law of thermodynamics, a law that often troubles physicists when they think about life. There is no problem. Schrödinger even had concerns about it. He discussed it quite a lot in his book. But he, and this is a nice sentence from Schrödinger's book, reflecting an organism, a living organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. He was very concerned about that, but there is no problem. It's separated from the rest of environment, and that allows order to take place. He was astonished by this fact, escaping the decay into atomic chaos, and that led him to propose that life had a code script. And these quotes introduce the second idea I want to share with you. You see this sentence here, these chromosomes, this was the 1940s and chromosomes were identified by then. These chromosomes contain in some kind of code script the entire pattern of the individual's future development and functioning. And this is explained by the concept of the gene, which is the second idea I want to talk to you about, because it explains the mysterious phenomenon of inheritance. This is the idea that the gene is the basis of heredity. And it had its origins with the work of the Augustinian monk Gregor Mendel, abbot of Brno Monastery, now in the Czech Republic, Česka, when he published 2 papers in 1866. By the way, you'll already have noticed that some of the fundamental concepts, I'm on to my second, are really quite old. One from the 17th century, and these two, and then made more precise in the 19th century, and this also from the 19th century. Now, what did Gregor Mendel do? He tackled the problem of inheritance by carrying out crosses of pea plants with different characteristics and analyzing the outcomes in thousands of progeny formed in these crosses. He was actually trained as a physicist in the University of Vienna. He never passed his exam, so he never got a degree. But he was trained, perhaps I should not say that in this audience, but he never got a degree. But he thought in a very numerate way, probably as apart from his training as physics. I found this picture of his garden, which is from 1981, when I visited the monastery in the middle of the winter, actually, and also the middle of the Cold War, as it turned out. It's very difficult to go there. And discovered all his artifacts locked away in a cupboard, where because of Lysenko-ism, which was still very prevalent in Eastern Europe of the time, they were being protected. It's now, if you go to Brno, you now will find all those exhibits which I was handling are now kept carefully in a museum. And what Mendel did was to cross peas of varied characteristics, which he could identify very clearly, plants of different heights, different flower colors, different seed shapes, and you can see examples of this here. And when he crossed them, he famously observed simple ratios in these characteristics in the progeny produced by these crosses. Ratios which we will have all been taught at school, 3:1, and 9:3 to 3:1, and 1:2 to 1. Clear ratios. This eventually led to the idea of Mendelism. Actually, in his papers, he didn't quite get to Mendelism. It's a little obscure, but within the next 30, 40 years, it did lead to Mendelism. And that idea is that heredity is based on pairs of indivisible particles or factors, one of which is inherited through each of the germ cells, pollen and ovules in plants, sperm and eggs in animals. And these factors were subsequently found to be linked to the chromosomes, already mentioned by Schrödinger, though 40 years later. But chromosomes were only discovered after Mendel's death, and nobody took any notice of Mendel's results in 1865, none at all. They were rediscovered in 1901, 1902 by 3 research groups. The first didn't really want to acknowledge Mendel. The second one, who was, of course, beaten to publication by the first, talked a lot about Mendel, so he could undermine the first one. And then the third put it all together. That's not quite true, but sort of half true. The chromosomes were discovered after Mendel's death and were found to be separating in dividing cells, as shown in this this is an original late 19th century drawing of onion root tip cells. And what this allowed is for Mendel's abstract interpretation to be concretely observed by observation. Abstraction is quite difficult for biologists, and they prefer concrete observations. And linking the abstraction of just numbers to something that was physical made it more acceptable to biologists. I have to say, I first saw chromosomes in an identical squash of onion root tips a few months after the butterfly incident. So it's rather close to my heart, these things. These Mendelian hereditary factors are what we now call genes. In the 1940s, Oswald Avery in Rockefeller University, New York, where I worked for 7 or 8 years, showed that genes were made of deoxyribonucleic acid, or DNA. Didn't actually get that much attention at the time, in 1944. Did subsequently, but not then. And here you see a nucleus being expanded, a chromosome, the chromosome being pulled apart, and now you can see from that the DNA molecule that you'll be all familiar with. The structure of DNA as a double helix was established 10 years later, in the early 1950s, in London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, who were all involved experimentally, and in Cambridge by Francis Crick and Jim Watson, who did the modelling. And they, that is Crick and Watson, famously argued that the double helical structure of DNA could explain heredity through the linear sequence of bases making up the DNA and the pairing between complementary bases, adenine with thymine, cytosine with guanine. The double helix is like a twisted ladder, is what you can see it here. I want you mentally to untwist it and just imagine it a ladder, okay? And what this means is that the rungs of the ladder are made up of the links between these complementing bases, and when the links are split, 2 templates are formed, which can be used to make 2 new identical helices. And this, of course, can explain how DNA is precisely copied during cell reproduction, accounting for heredity. It explains Schrödinger's code script and the permanence of the gene between generations, because DNA acts as that code script precisely copied every time a cell divides. And as you heard in the introduction, it's how all those processes during cell reproduction has been the center of my research life. Crick went on to propose what is called the central dogma, explaining how DNA encodes the structure of proteins. The central dogma describes how the sequence of bases making up a gene is copied from the DNA to ribonucleic acid, that's messenger RNA. That acts as a messenger from DNA located in the nucleus, where the chromosomes are, of course, and the cytoplasm, where proteins are made. And in principle, there's one gene encoding each protein. These discoveries, which were brilliant, laid the foundations for molecular biology. They are critical for understanding life, absolutely critical. Central to this is that genetic code. Heredity is written in this 4-letter code made of the 4 bases, adenine, thymine, cytosine, guanine, with the information stored in a linear form. This way of storing information is the same as the sentences I am speaking to you, or the sentences you read in a book, or bytes in a computer. It means that the linear digital storage of information was invented by life over 3 billion years before the computer age, just to put us in perspective. The nature and function of the gene introduces the next two ideas I want to discuss with you: life as chemistry and life as information. The function of living organisms is based on chemical activity and physical forces. The origins of this idea can be traced back to Lavoisier, the French father of chemistry, although he lost his head in the revolution, in the guillotine, so didn't have time to fully generate his ideas. So it was picked up by another Frenchman, Louis Pasteur, who carried out experiments on sugar beet fermentation, which led to the concept that life is based on chemistry. This is a romantic picture of Louis Pasteur, which is in the Pasteur Institute Museum. Pasteur was analyzing the fermentation of sugar beet to make alcohol at the request of industrialists in the northern France, because the fermentations sometimes failed. And instead of making alcohol, which they could sell, they made acid instead, which they couldn't sell. So it was a commercial problem. And Pasteur demonstrated that the successful production of alcohol required the activity of a single-celled living organism, that is, yeast. He proposed that the yeast cell produced substances that made alcohol from the sugar derived from the sugar beet. And if yeast was not there, then there would be a contamination by bacteria, and the fermentation made acid instead. And so that's why it was ruined. But Pasteur did more than that. I mean, he obviously solved the problem for the commercial people. I don't know if he got paid for it. I haven't been able to find that out, but I'm sure it made him popular with his colleagues there. But Pasteur did more than that. He concluded more generally, and I quote, again, a translation from the French, that fermentation was a physiological act yielding products for the cell. That is, chemical reactions are an expression of the life of a cell. These are beautiful summaries of what actually is true. Mid-19th century, once again, ideas that are 150 years old. And in short, what that means is life is based on chemistry. But it is a very elaborate and rather special chemistry. It's based on carbon polymers. DNA and RNA are both carbon polymers. And now we need to think of the carbon polymers that make up proteins, because these are the substances from yeast that Pasteur postulated made the alcohol. They act as enzymes which catalyze chemical reactions that make that alcohol. So what are proteins? Proteins are polymers of amino acids with a backbone and side chains from of the different amino acids. Here is an example. You'll see a backbone which is made up of carbon, carbon, nitrogen, carbon, carbon, nitrogen, each of which come from a particular amino acid. But there's 20 amino acids, and they have different side chains, which are shown here in color. Lysine, leucine, tyrosine, aspartic acid. And those different side chains have different chemical properties. Lysine is basic, for example. Aspartic acid is acidic. Leucine is hydrophobic, and tyrosine actually is rather bulky and has got a cyclic arrangement. So these different amino acids have, as a consequence, different chemical characteristics. And these are only examples, the ones shown here, of 20 different amino acids that make up proteins. This is another picture of the same structure, but actually with the lengths of the bonds and the nature of the bonds. But these amino acids make up generate a different chemistry which can carry out many and different chemical reactions. This contrasts with DNA, because frankly, DNA is very stable and rather boring. It's not chemically diverse, it's stable and rather dull. But the chains of amino acids in proteins result in the protein polymers folding up in many different ways to make complex molecular structures. Here is a chain of a protein. You can see the different amino acids. And this is when it's folded, and you can see each of those balls is an amino acid with a different chemical characteristic shown by the color. And these varied structures, combined with the varied chemistries of all those amino acids, accounts for the wide diversity of chemical reactions that enzymes can carry out. So enzymes and other proteins, like enzymes, act as chemical machines. They produce a huge range of chemicals required for living organisms to function. They make new molecules, they break molecules down, they recycle them, they make large molecular assemblies, they make nucleic acid, proteins, lipids, carbohydrates. They can do physical work, they make motors, they carry cargoes around on protein-based trackways. They act in hybrid ways of chemistry and physics, capturing energy from sunlight to produce energy-rich chemicals that can drive activities in cells. All this means is that that simple little cell that I showed you earlier, within it, thousands of chemical reactions are being carried out simultaneously, together, very close together in living cells. A small sample of the reactions going on is shown here, and there will be possibly some of you old enough in the audience to have remembered learning this. This is a map of the central metabolism of cells, the chemistry of life. And you'll see that circle of red, that's the Krebs cycle, if you're interested. The department I was in Oxford used to be headed by Krebs. Now, because these reactions require different chemical conditions, they have to be separated one from another, otherwise you won't be able to get the varied chemistry. This is achieved by compartmentation, brought about at various levels, brought about by the surfaces and folds of enzymes, which can make little pockets, by complexes of enzymes acting together, by membrane-bounded organelles. Back to the cell, that's what actually the cell is you are seeing here. In fact, the way you want to think about this is not just as a static picture of the cell, but as a way of generating separation of chemistries into different parts within the cell. And this allows the hugely elaborate chemistry of life to take place. It is completely extraordinary. Man-made commercial productions have to produce large chemical plants in extreme conditions. And here we are seeing hundreds, if not thousands, of reactions going on simultaneously in this tiny space. It is extraordinary, it is wonderful, but it is actually what is happening. However, it's even more complicated. For this to work, all this chemistry must work together as a whole with a purpose. It's not just random reactions. They have to be integrated in what they're doing. They have to be organized together. They require communication between these different chemical microenvironments and compartments. And communication involves the transfer of information. And that leads to my next idea, life as information. Now, life is a complex system. I wanted to find out where that was first discussed, and you won't believe what I'm going to tell you. The first place I've traced it back to was the philosopher Immanuel Kant. And Immanuel Kant, in a book he wrote on moral philosophy, has about 20 pages on the complexity of life. What on earth it's doing in a book on moral issues, I have not the slightest idea, but he did. Of course, he didn't explore it much further, but it is where I first came across where it was discussed. Now, the operations of such complex systems are based on the management of information. And I'm going to give two examples to illustrate this. The first is the structure of DNA. This you'll be familiar with. The structure of DNA, the double helix, is iconic and it is beautiful. But its real beauty lies in the fact that it only makes sense, biological sense, when it's recognized as a digital information storage device, as I've already briefly mention. One written in that linear script of 4 letters. The linear polymer of DNA is ideal for storing information, with the encoding letter bases turned inside the helix and protected from chemical change, which is what is required. But this information's got to be turned into chemical work, chemical action. So that's separated by it encoding proteins, which can then carry out the chemical work. And for me, this is a wonderful characteristic of life, all built on carbon-based polymers. The ability to encode information in inert one-dimensional linear structures and to turn that into chemical and physical work in the production of proteins in active three-dimensional chemical structures. Not emphasized enough in our biochemistry textbooks, I would suggest, but it is a wonderful characteristic. Second example about complexity, information, and control I want to tell you about is about regulation. That is making the complex systems in living organisms acting as a whole with purpose. A familiar example of regulation is found in man-made machines, and we can more easily see that if we look at man-made machines. Now, I don't know if you're like me. I understand most machines made before 1900 and almost increasingly none after 1900. So what I'm going to show you is a picture of a governor on a steam engine. I took this a picture. I found this engine in a New Zealand ferry boat built in 1890. And as the engine goes faster, the balls swing out, and that shuts off steam through a valve. And when slower, they swing in and switch on steam through it. So we can sort of get it, okay? And this leads to what we call homeostasis, maintaining a constant speed of the steam engine without a person having to control it. And the governor, as it's called, which, by the way, was invented for windmills before steam engines, but was adapted for steam engine. The governor is an example of a negative feedback loop. That's shown here at the top of this logistical diagram here. If we look at that, it's a metabolic pathway. It goes from, has A, it turns it into B, and turns it into C. As C builds up, it inhibits A to B, shutting down the synthesis of C, leading in the homeostasis in the level of C. Very straightforward. Yes, everybody, I'm sure, got that. Negative feedback in cells was demonstrated by the great French, brilliant, actually, French geneticists François Jacob and Jacques Monod, studying gene regulation of sugar metabolism in bacteria. Other control models lead to different outcomes. A second one, there's a range of them, is a positive feedback. And in this one, as C builds up, it accelerates A to B, making more C. So this acts as a switch. Once it happens, it's irreversible switch. So you can go from homeostasis, you can go to switches. Many such feedbacks and control modules operate in cells, maintaining homeostasis, switches, timers, toggles, oscillators, translating chemistry into informational modules. Chemistry into informational modules. And these can be linked together to generate even more elaborate routines of informational management and processing. A metaphor for all of this is an electronic circuit of linked modules. And this I stole from some journal. It's a transistor radio, and those of you who work on cancer will recognize many cancer genes here linked together in hardware in the transistor radio, a metaphor for what happens in cells. But an even better metaphor for living systems is not hardware, it's wetware, because actually the components are linked through solution chemistry, and that means they can be re-linked in different places. And that is something we've barely explored, but there is evidence for it, and that it has the potential to greatly increase the versatility of control modules. So the management of life's chemistry by information allows the behavior of a complex system to act as a whole, with overall purpose. I'm using the word deliberately purpose. It's not purpose like a purpose in the human brain. It's simply leading to outcomes of a whole rather than individual components. For centuries, the generation of purposeful behaviors was thought to require a designer, a divine creator. And it took evolution by natural selection to change this. And this is my final and fifth idea. So if you're getting a bit weary, this is the last idea. You can relax, have a deep breath. We're getting towards the end, okay? Evolution by natural selection. How living things come about, or at least develop. This is a beautiful idea, largely due to Charles Darwin. But the idea has two parts. The first is life evolves over time, which was not Darwin's idea. That had been thought about before. And secondly, which was primarily Darwin's idea, a major mechanism for evolution is natural selection. This is a picture of Charles Darwin, the one on the right. The man on the left, I want to say a few words about. That was Charles's grandfather, Erasmus Darwin. And that life evolves was widely discussed in the century before Charles, particularly by Jean-Baptiste Lamarck, for example, who made many contributions, and Charles' own grandfather, Erasmus Darwin. Erasmus was a really colourful character. He was a scientist, a doctor, a poet. All his research is written in blank verse. I collected his books, which are books of poetry describing what he thinks is going on. He was a Republican, a proponent of public women's education, and wrote a book on that subject, and a great conversationalist, despite having a terrible stammer. I love him. He's obviously really interesting character. Now, the evolution of life, to go back to Charles, it implies there's a tree of life. That is, living organisms are related in a tree of life. And if life only arose and survived once, and I think that's probably likely given the conserved chemistry and coding found in all living organisms, then that means all life on Earth is related to each other. Quite a fundamental principle. And this connection of life was captured in the only diagram in Darwin's Origin of Species, published in 1859, where he illustrated the first tree of life. And you can see that the branching tree with animals and plants going extinct. At the top we see the organisms that have survived. But look at the bottom, the A to L. He was open as to whether life arose more than once and entered that as a possibility. Charles Darwin gathered evidence in favour of life evolving through natural selection during his trip around the world in the Beagle. Let me explain natural selection briefly. It's a consequence of populations of a species exhibiting variations caused by inheritable genetic changes. These can be caused by mistakes when DNA is copied or if DNA is damaged. If these variants influence characteristics that make these organisms more successful, better adapted, then they will reproduce more, and that genetic variation will spread through the population. And over time, such genetic variations will accumulate and lead to changes in the population, such that individuals of the new population will no longer be able to breed with the original population. That's 5 sentences describing Darwin's Origin of Species, because this is the origin of species. But he took 400 pages, of course, because he explained it better and had evidence. Darwin came to this view in part by his studies of the beaks of finches on the Galapagos Islands. I stole this, I forget, from probably Scientific American. On the left, you see Darwin's finches. On the right, you see lots of pliers. The birds' beaks, the big ones can break nuts, the thin ones could catch insects under the bark of trees. They became adapted to their lifestyle, their environment. They became specialized better for what they had to do to succeed. Pliers are exactly specialized in the same way to do different jobs. They have the same basic structure, but they're specialized to do different jobs. But the big difference between beaks and pliers is that pliers are intelligently designed by human beings for different specialized purposes, whilst the finch's beaks achieve specialized purposes by natural selection without a divine designer or a creator. And that is the nub of the argument of the differences of the creationists who believe in that sort of design, intelligent design. Now, for natural selection to take place, three things are needed. One, organisms have to reproduce, okay, clearly. Two, they have to have a hereditary system. Three, that hereditary system has to exhibit variability upon which selection can work. These are actually based on two ideas I've already described, the idea of the cell and the idea of the gene. Imagine a single-celled life form which has got a brown coat. Imagine that the color of that coat is determined by a gene. Imagine that a mistake occurs in DNA so that the coat becomes red rather than brown. And also imagine that that red cells survive better, perhaps because they avoid predators. You can make up whatever you like of it. As a consequence, the red cells replace the brown cells in the population, and evolution by natural selection has taken place. That's the basis of natural selection. This is a simple but profoundly important idea in biology. So much so that the 20th century geneticist Hermann Müller used it to define life. He defined life as living things which have properties which allow them to undergo natural selection and therefore evolve. And that's going to be an important part when I finally summarize at the end of my talk, which is in a couple of minutes. But before I do that, I want to say goodbye to Darwin as an old man. This is a picture as the old sage with the final monumental sentence from his Origin of Species. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. Why can we not write sentences like that today? I write a paper, I can't bear to read it after 3 months. By the time it's gone through editors and by the time the reviewers have taken out anything of interest, it's complete dross. And you can read this sentence from 1859 and feel motivated, feel inspired. It's not all progress, it's not all progress. But notice there, he was cunning. He was relating to the great physicist Isaac Newton with gravity. And what he was really saying, I think, with this sentence is that biologists can have great ideas just like the physicists can have great ideas. And that otherwise there's no reason for mentioning the law of gravity in a book of, on evolution. Now, I'm about to define life. But I first need to consider the difficult category of intermediate life forms, particularly viruses, which always, the school essays, is a virus alive or is it dead? Viruses have a genome, could be DNA or RNA. They undergo evolution by natural selection, as we see with COVID, for example. They pass, therefore, Muller's test, but can only reproduce when they're inside the cells of other host living organisms by hijacking that cell's molecular machinery to copy its genome and to make more viruses. And this means that a virus is completely dependent upon another living entity. You could also sort of say a virus is alive when in a cell and a lifeless chemical when outside a cell. That's another way of doing it. But the question is, are viruses truly alive? And I want to say something to try and weave through this. In thinking about it, it's important to remember that other forms of life are also, to greater or lesser extent, frequently very dependent on other living beings. The many parasites that live on or inside cells or bodies of animals, plants, or fungi are, for example, examples of this. The extent of these dependencies are less than, of course, for a virus, but they are still dependent. There are some amino acids we cannot make and are made by microbes that we have inside our bodies. Other organisms we use as food, so we are dependent upon them. Therefore, there's a graded spectrum of some living things which are highly dependent, such as the virus, to others which are only partially dependent. But I argue that we need to consider that, but these different life forms are all alive because by Muller's definition, they all evolve by natural selection. It also means, the Darwin's fifth idea, that life on Earth is fundamentally connected, both through being related as a result of evolution, remember they're all our relatives, and also through these deep relationships and interdependencies. Okay, I said I'd bring this together. I'm now going to summarize where we've got to on these slides. Living things are independent, bounded physical entities. This escapes computer games. The basic unit of life on Earth is the cell. They're separated from their environment. They're in communication with it. This allows order to be generated. Multicellular organisms, like our cells, are aggregates of cells. Living things possess the ability to evolve by natural selection, and to do so they have to grow and reproduce and have a hereditary system which exhibits variability. 3, living things are chemical, physical, and informational machines. They construct their own metabolism, and they use that to maintain themselves, grow, and reproduce. And on Earth, life is based on carbon polymer chemistry. They produce nucleic acid information storage devices and protein chemical and physical machines. Life is based on information management, and living things on Earth are related by common descent. Now, should we ever encounter life elsewhere in the universe? I suspect it will be built on similar fundamental principles. The chemical details may be different. Perhaps it's not even based on carbon. The science fiction writers like silicon quite often when they think about these things. However, to satisfy the need for long-term information storage, I think it's likely the chemistry will always be built on polymers. And I think we've underestimated the importance of polymers in thinking about the nature of life. But to return to our planet, because it's the only corner of the universe where we know for certain life exists. The life that we are part of here on Earth is very diverse, but we are beginning to make sense of it, and that growing understanding has potential to improve the lot of humankind. But that knowledge goes further. Biology shows us that all living organisms we know of are related and are closely interacting. We are bound by a deep connectedness to all other life, to bacteria, fermenting yeast, pea plants, to yellow butterflies. Together, all these species are life's great survivors, the latest descendants of a single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. I want you to note that sentence. We are connected through cell divisions, and it goes back and back and back to 2.5, 3 billion years. There's a sort of physical connection to all life. I mean, till I wrote the book, I didn't really think about that. But as far as we know, we are the only life forms who can see this deep connectivity and perhaps reflect on what it means. And I argue that gives us human beings a special responsibility for life on this planet. It is made up of our relatives, some close, some distant. We need to care about it, we need to care for it, and to do that, we need to understand it, which is what I've tried to do in this talk. Thank you very much for listening to me.
Thank you, Sir Ponners, for the inspiring lecture. Please remain on stage and take a seat for the upcoming Q&A section. Just now, in the thought-provoking lecture, Sir Ponners explored the profound questions, what is life, through the lens of 5 fundamental concepts of biology. During the next Q&A section, Serponers will embark on the enlightening conversation, illuminating the definition of life with Professor Dennis Low. May I now invite Professor Low to come on stage to moderate the section. Professor Low, please.
So thank you very much. That's really a very elegant summary about the essence of what it is to be alive. Actually, before I open up the session for questions from the floor, towards the end of your talk, I was thinking about something else. I was thinking, now, let's say that if eventually we develop artificial general intelligence, and let's say that if that AGI have access to, factories, robots, and they can build itself, right? You can build something, and then it learn, it see the environment changes, so it adapts in a way, almost like the evolution. Then in that sort of system, it seems to be, and it's very similar to life, and yet quite different from what you told us. So what do you think about that?
Well, I think it's quite difficult to achieve that, but let's say one could theoretically achieve it. Then, and I think we have to philosophically think carefully about whether we should think it's life. Because I ended the talk by saying we might have life in other places in the universe built on different chemistry and perhaps different ways of putting things together. And just maybe, if it was exactly as you said, because I avoided computer games and so on, which can also evolve because it's dependent on something else. But if the whole thing can organize itself to have robots, make the robots, and somehow evolve. I'm not quite sure how natural selection works in this particular situation. Let's say you could get it to work, then I think we should consider it we should need to consider it seriously. I think it's unlikely, but not completely impossible.
Thank you so much. So any questions from the floor?
I hope they're not as difficult as that one, though.
Yes, please, Albert.
I support this was fascinating talk. I want to talk a little bit something slightly different. You talk about chemistry, biologies, and it seems to me like this is a stew of a soup of chemistry of a human being. As a skeptical agnostic, I want to hear your view about whether or not there's something more profound, bigger than this.
You mean in human being aspect?
Yeah. Yeah.
Well, of course there is.
Life.
Of course there is. You'll notice how I defined the problem, because I do not consider issues like consciousness and other aspects of which you could say are more important than just life itself. So I restrained my consideration to living things as a whole. And that's as far as I feel able to go. But when we think of a human being, you have living cells inside you, but a living cell is not you. It could possibly produce you, but it is not you. Because when it produces you, it acquires which is what happened to you when you were a fetus, and I'm pointing at you a lot just to sort of make it real. You produce emergent properties, essentially, that lead to behaviors, consciousness, and all sorts of human characteristics. Now, there is a part of that in even quite simple organisms which are aware of their environment, and they are functioning, and they're also self-aware, even a cell in a sort of way. But it is not the way that you're describing the nature of a human being. So there's a whole new book to be written, as many books have, which is the emergent properties of the human being. I don't consider that. I think it's really interesting, but I've only written about something I know about, and I don't know the answers to those questions.
Thank you. Tony.
With your great talk and a great topic on what's life, I cannot stop myself to ask the question, what is death?
Well,
As a simple doctor, we just say when the heart stops pumping, that is death. But with your description of the different functionality, should we wait a bit longer before we pronounce someone dead?
Well, I have to tell you, I'm preparing a second edition of this book, okay, and one of them I'm having to think about what is death, because people have asked this exact question. And it amounts to the fact, of course, that there's death of a cell, the individual component, and there's death of you, okay? And the sort of life that a cell has is killed when the cell dies. The sort of life that you have is partially killed when certain elements of your body become defective. Now, I've had a heart bypass, and they stopped my heart for several hours to do that. And so my heart stopped, which would normally, but I was kept alive, of course, as you would know, because I'm still here and still talking to you, and as a consequence of that, survived a process. So we can substitute certain organs, sometimes temporarily, sometimes by transplantation, but we had to be aware of that. And now you ask the question, when is somebody dead? And we sort of go back to thinking a lot about the brain and whether you've got perception or awareness. But increasingly we're finding, as you I'm sure know, by looking at brain behavior, that there sometimes is in fact some function there when people have been thought to be brain dead. And I think this is really a complicated issue. The principle here is, though, it is overall behavior of the whole that you're looking at. And if the whole is defective, then you may not be still alive. But there are some aspects of the whole which can be defective. If I lose a leg when I'm clearly alive, and there's others that take you close to a boundary. But it is clearly a very important problem for a doctor like you to know whether somebody's dead or not. Yes, maybe.
Thank you. Any other? Yes, there's a gentleman in red at the back, please. So Edwin.
Thank you, Sir Paul. Very exciting talk. And I'm a teacher from the School of Life Sciences. I've been thinking about the limit of life. And would that be limited by the central dogma, or for example, the amino acid combinations of a protein, or limited by the 20 amino acids composition of a protein? Say, for example, we can easily change a thermophilic protein to a mesophilic protein. Can we go beyond the thermal limit? Say, for example, the aqueous environment changed to an organic environment. Can we still survive with the central dogma that we exist or we rely on these days? Thank you.
Yes, it's an interesting question. Let's confine ourselves to cells, if you don't mind. I mean, because limits of human beings is again somewhat complex, and it is clear, at least I think it's clear, that the basic chemical structure of life as we know it has certain limitations. Sometimes we've been wrong about those limitations. So, for example, at the beginning of my career, we didn't think living organisms would survive at high temperatures, and now we know that actually they can survive certain microbes at extremely high temperatures. So certain limitations that were thought to be sort of universal for life are not universal for life. But there's bound to be certain limitations. So the fact is life might be able to live at 60, 70, 80 degrees centigrade, but life as we know it is not going to live at 500 degrees centigrade. So there are certain boundaries which are a consequence essentially of the chemistry, I would say. Within those boundaries of the fundamental chemistry, I could imagine new networks evolving that could give new capacity that we haven't thought of. I mean, because remember, life as chemistry, life as information. So the way everything is connected, I think there is an enormous variety of possibilities there, which could lead to new ways of behaving. And in a way, synthetic biology could begin to address that. And we may be able, through synthetic biology approaches, changing the types of amino acids that might give new sorts of chemistry. So I think all of that is could further the limits of life, but there will always be limits. They just may change. Once we take your question and broaden it to, humanity, to human beings, the whole thing gets really a little more complex, and it does tend to go into science fiction. Thank you.
Yes, this gentleman on the left, yeah. Okay.
Hello, Sir Paul. Thank you very much for your inspirational talk. And well, I got a question for you. Actually, two questions. Well, first,
one at a time. My memory is not.
Yeah, of course. So when did you discover your passion for working on eukaryotic cell cycle? I mean, what is the motivation behind?
Yeah, no, thank you for asking that. I was doing my PhD, and being a researcher is a miserable business. You see, most experiments fail a lot of the time, and what I thought was, look, if I'm going to continue this, knowing that often we fail, at least I should address something I think is important. Okay, and I thought the reproduction of a cell from 1 to 2 is a basic property of life, reproduction, and that's worth investigating. I also thought that working on the cell cycle, which is what that process is universal to eukaryotes, certainly what I was looking at. And that even if I failed to succeed, at least I was trying to succeed something that I thought was important. So I think that's my short answer. What's your second question?
Yeah, thank you very much. So the second question would be, forgive my curiosity, but how did you or when did you discover? Because the topic of today is what is life. Our mothers gave life to us, gave birth to us. So I wanted to ask, how did you discover that your older sister was actually your mother? I mean, that process must be quite shocking to you, I'm sure.
Sorry, I didn't catch. I
was asking, how did you discover that your older sister was in fact your mother?
In fact, sorry.
So,
forgive me.
He's asking about the story about you discover your heritage.
Oh, mine, yes.
Forgive my curiosity.
Oh, yes. What's being referred to is that I was brought up in a family, and I only discovered in the 50s that my mother was actually my sister. And I was brought up by my grandparents. This is what you're referring to. I was brought up by my grandparents pretending to be my parents. And I found out in a curious way, because I had moved to the United States and my request for a green card was rejected. And since I'd written about 20 references for other people to get green cards, I was a bit actually surprised about that. And it was because they didn't like my birth certificate. It didn't name my parents. And so I asked, it was a short birth certificate, which was legal. And I'd asked my parents why I had one, and they said it's because it was cheaper than the big one, okay? And I believed them, but it wasn't true. It was to hide illegitimacy, because I was illegitimate. So I discovered my mother was my sister. And she had died by then, so I had oh, by the way, the birth certificate did not name my father. Did not name my father. And I only discovered who my father was about 2 years ago, when I did one of these genetic tests, and I traced it. Well, I didn't trace it back personally. I tried to contact the people who connected, and they didn't reply. And being British, I didn't want to sort of upset anybody, so I sort of just, let it alone. And then I was on a radio program, and this came out. And the person who was with me was a genealogist, geneticist. Her name's Thury. And she'd done Richard III. Do you remember any of you Richard III being dug up in the car park in Leicester? And she said, maybe I can help you. And that was on a Friday. I remember giving her the information to get access to my DNA. I was in Terminal 5 in Heathrow, that somebody would know. And I went to Japan. I was in Hiroshima. And on Sunday morning, she emails me and says, I think your father is X. You know? And she was right. She was right. I had done testing. That was only 2 years ago. So I was curious, like you, who my mother and father was.
Thank you for sharing that. So, okay, now, so maybe this one is Ben, right? Yeah.
Thank you very much for your talk. In your answer to what is life, you start by emphasizing the independence of living things, and in your final part of the answer, you highlight the interdependency between different aspects of living things. I wonder, to what extent do you think that Eastern cultures, philosophies, history of science might help to strengthen that aspect? Because a lot of the Eastern ways of seeing life, whether it's Buddhism or other philosophies, do emphasize that, and many of the examples you refer to are more from European history of science and so on. So I wonder whether that's something you're exploring.
Well, I think in fact, all the ideas I spoke about come from Europe. I mean, and I emphasize how, I just repeat, how quite old some of them are. But the interdependency where I finished was more my own design. And of course, it does, it sort of vibrates with some Eastern thinking as well. I'm sort of aware of that. What I sort of think about generally about science and investigations is you have to have an open mind to address all types of thinking, and it will actually assist you to come to solutions. And sometimes we're just too limited in the way that we actually think about problems, which I suspect is what you think, given the question.
Thank you.
David Foster.
Thank you, Sir Paul. It's a really inspiring talk. Just building on this a little bit about some of these ideas being very old. We also saw the equipment was quite old, but still we saw the garden that Mendel used was quite small, surprisingly small, and we used the microscope that Hooke used, very basic. Do you think that now science has become too specialized? We've reduced the level of wonder because three of the concepts, the Hooke, the Mendel, the Darwin, the finch's beaks, and the cell, these were identified using very, very basic equipment, more or less like the regulator that you showed us.
Yes, actually, I think the garden was rather more elaborate than I expected to find in a monastery. It was actually in a monastery. He was a monk. And that picture of the garden, he had adjacent to it a greenhouse of similar size. And I was just astonished that a monastery would be supporting a monk to carry out these experiments in the middle of the 19th century. I mean, it is fairly astonishing if you think about it. But the principle stands what you said. Of course, it's very simple what they had. I mean, this is interesting, isn't it? I mean, you know, when Galileo used a telescope to look at the moons of Jupiter, people were deeply suspicious of it because the telescope got in the way between the human perception and the object, and they saw that as a potential risk that your perception will be distorted by the, in principle, instrumentation that you're using. Now, that applies even more today, and the point you're making is that we're made a bit remote from what we're studying by the very methodologies we have to apply to get any access to what's going on. So we have to, that means we have to be really careful about the instrumentation we use. And we can get a bit blasé about it. You know, we have good resources, instrumentation resources. I mean, I saw some excellent ones earlier today in a very sophisticated laboratory. The Francis Crick Institute, which I established very good technical cores. But the reality is, my students, and for that matter myself, are no expert in all of these things. And we have to be very, very careful about how we progress, because I think we can, we're not always understanding the methodologies that we're using. It's a little bit like as a metaphor for that, because I think I'm going on about this because it's an interesting question. You know, all my students now do statistical analysis because you can press a button and it just happens. They don't sort of get it, whether it, you know, quite what it means. They can just do it, and they've become remote from it. So that's one sort of problem. The philosophical point, which I think your question also touches upon, as to whether we get seduced by the analysis and the instrumentation and the fact that we can describe many things and lose the point of what we're doing. Now, I'm going to be rude now. There's an institute in Boston called the Broad Institute, and I think there's a disease I call broditis, okay? And the disease, broditis, is that you publish a paper, usually in Cell, by the way, which is an enormously, got a fancy setup, a very fantastically complex analysis, and comes to almost zero interesting conclusions, okay? And this is a caricature, of course. Is this being filmed, by the way? Anyway, but you see the point I'm making. Sometimes we need simplicity to think about the important problems. Sometimes we need time just to think about it in terms of being close to the biology, and not to be seduced that that if we can measure 5,000 proteins, it's enough to measure 5,000 proteins. We have to have a reason for measuring 5,000 proteins, of which there are good reasons in certain circumstances, but we need to be close to the biological problem that we're trying to address. So I think I have some sympathy with the position I think you're arguing, but I think there is an issue we have to pay attention to.
Thank you. Now, I think we can just take one final question. So, Kathy.
Hi, Paul.
Hi.
That was very inspiring. I'd like to go back to the fundamental concept that you were talking about, and that is the concept of the cell.
Yeah.
And I wonder what you think about even in unicellular organisms, whether it is essential for life to have cells communicate with each other, because at the end of the day, even a unicellular organism doesn't actually exist all by itself. So if cells could not communicate with each other, do you think there will be life?
No, because it couldn't evolve, or it would evolve extremely slowly. Single-celled organisms, as you know, mate with other single-celled organisms, and it's part of the mixing up the genetics that allows evolution by natural selection to occur. You can, of course, get evolution by natural selection without that, but it's very, very slow, and indeed. So I think single-celled organisms nearly always do communicate one with another so that they can mate with each other, and there's a reason for it, which is genetic diversity, which is needed for evolution by natural selection. Single cells behave purposefully, which isn't quite the question you had. So there's other ways of which they communicate one with another when they're growing together and so on. So there is other things which do require communication. But it sort of means that aspect of communication is something which single cells have and is elaborated to a much greater degree in complex organisms, but is there, which I think was the basic question. Wait, can you wait for the mic? Because I'm...
So communication within between cells.
Between cells, yeah.
Is very essential then, you think?
I
for driving diversity.
Okay, let's be more specific. Communication within cells is absolutely essential.
Of course, yes, between
absolutely
cells.
essential. It's only another step to get communication really between cells. And communication between cells brings more to the party that you need to be successfully alive. Although one could imagine life without it, it would evolve very, very slowly. I think I've never been asked that question before, so maybe there's more to be said.
I mean, in terms of driving diversity, right? I mean, because we have different species, we have different forms. You've really, illustrated that beautifully. But to what extent does that depend on the ability of cells to communicate with each other that leads to diversity? Because also the signaling pathways are conserved, but not always, right?
The diversity is really, as I'm answering it, and as I said, I've not really thought about this before, is all to do with the mixing up the genomes so that you can get much more rapid variants. If you are just reproducing vegetatively, you just rely on single mutations for adaptation, and it could occur. But if you can mix them all up, it's much richer.
Thank you. So on that note, so let's draw this discussion session to a close, and let's thank Sir Paul again. Thank you.
Thank you, Sir Ponners and Professor Low. Please remain on stage for the next section. As a small token of appreciation, Professor Low would like to present a souvenir to Sir Ponners. Professor Low, please. Thank you both, and please remain on stage. It is my great pleasure to invite Professor John Chai, Chairman of the Council, to come to the front for a group photo. Professor Chai, please. Thank you all, and please remain on stage for another group photo. Now, please join me in welcoming the guests from the first row to the stage for a group photo. Dame Julie Maxton, Executive Director of Royal Society. Professor Michael Hauser, Fellow of Royal Society and Director of School of Biomedical Sciences, the University of Hong Kong. Mr. David Foster, Executive Director of Croucher Foundation. Mrs. Susanna Morley, Director of British Council, Hong Kong. Professor Sham Mei Ha, Vice-Chancellor, Vice President, Research of CUHK. Professor Anthony Chan, Vice-Chancellor, Vice President, Alumni Community Engagement and Institutional Advancement of CUHK. Mr. Albert Wong, Council Member of CUHK. Ms. Lena Yen, Council Member of CUHK. Professor Erwin King, Vice-Chancellor, Vice President, Education of CUHK. Professor Jiang Li Wen, Vice-Chancellor, Vice President, External Affairs of CUHK. Please look at the camera. Thank you all. Please be seated and join the audience for one last group photo. To ensure that the photo could capture everyone, may I invite the guests seated on the side, please move closer to the center, please. Everyone, please face the photographer in the center. Thank you all. Ladies and gentlemen, that concludes our event today. Let's give a big applause again for Sir Porners for bringing us such an exceptional public lecture.